Bismillahirrahmanirrahim. Assalamu alaikum, dear students. I hope everyone is fine and enjoying a good health. I'm Asma Kamran. Today we are starting a new chapter, chapter 6 in your book, Prokaryotes. We already have done that in chapter number 1. What are prokaryotes? Pro means first, karyotes is from karyon, means organisms which have primitive type of nucleus in which chromatin material or chromosome is not bounded by any nuclear membrane. They lack cell organelles, only ribosomes are present and are unicellular. These are different types of prokaryotic organisms, commonly called bacteria. The objectives of today's lecture is introduction of prokaryotes, taxonomy of prokaryotes, means classification of prokaryotes, Archaea, what are its features, how it is different from bacteria, and the types of Archaea. Bacterial ecology and diversity, Cyanobacteria, means blue-green algae, structure and composition of bacterial cell envelope, diversity in size of bacteria. What are prokaryotes? You already know what are prokaryotes, and they are very important because they have an intense effect on human health, economy, and environment. And nowadays, prokaryotes are extensively used in many different fields, especially in research and technology, and especially in biotechnology. Now, what is the taxonomy of prokaryotes? Means how we classify prokaryotes. Prokaryotes or bacteria was discovered in 1674 by Leeuwenhoek, and he called them animalcules. Animalcules means small animals. Ehrenberg introduced the term bacterium in 1882 from a Greek word bacterion or bacteria, means small stuff. The taxonomic position of prokaryotes has been continuously changed since their discovery, and the two separate systems of classification are followed in which the prokaryotes are accommodated in different ways. And taxonomic position of prokaryotes as kingdom, Monera. Previously we used two kingdom system in which all organisms or living organisms were divided into two major groups, animal kingdom and plant kingdom. All microorganisms, including prokaryotes, they were placed in kingdom, plant kingdom, because they are autotroph and they have cell wall. John Hogg in 1861 proposed kingdom Protista to accommodate microorganisms. Ernst Haeckel made Monera a separate group within the kingdom Protista in 1866, and Herbert Copland, he elevated the group Monera or Monera within kingdom Protista, or he elevated this group to a kingdom in 1938. Robert Whittaker proposed 5-kingdom system in 1969 on the basis of prokaryotic and eukaryotic distinction, that is on the difference of prokaryotic cell and prokaryotic cell. He divided organisms into 5 kingdoms. Lynn Margulis and Schwartz modified the 5-kingdom system proposed by Whittaker in 1988 and assigned a separate kingdom to prokaryotes called Monera. Nowadays, all organisms are divided into 3 domains: bacteria, archaea, and eukarya. Now, what is domain? Domain is a highest rank in taxonomy or in classification. It is a level higher than kingdom. Bacteria, archaea, and eukarya, they are classified on the basis of structure of cell and the genes that code ribosomal RNA. Bacteria and archaea both are prokaryotes, have prokaryotic cell, both are unicellular, but there is a slight difference in their ribosomal RNA. Eukarya, they have eukaryotic cell, and they are autotroph as well as heterotroph, have a definite membrane-bounded nucleus, have all organelles, so they are different from bacteria and archaea. Archaea were previously called archaeobacteria. Archaea means ancient. They are just like bacteria, prokaryotic cells, but are different because they are present in extreme environment where other living organisms cannot survive. Now, the phylogenetic position of prokaryotes. The evolutionary history of species or groups of related species is called phylogeny. In simple words, this means ancestral history. In 1977, it was recognized that Archaea has a separate line of descent from bacteria, means Archaea evolved separately from bacteria, although both are prokaryotic, unicellular, are similar to each other, but it was recognized that Archaea evolved separately from bacteria. This new phylogenetic taxonomy, it was based on the discovery that genes encoding ribosomal RNA are the ancient and they distributed all over lineages of life with little or no change. Means the genes that code ribosomal RNA are the most ancient genes, and from ancestral organisms to the modern organisms, they are distributed almost in the same way or in a similar manner, and they have little difference, or they change very little from ancestors up to the modern organism. So that's why organisms nowadays are classified into three major domains on the basis of the genes that code ribosomal RNA. So ribosomal RNA, they are recommended as molecular clock, and they are used for the construction of phylogenies, means ancestral history, and on the basis of this, organisms are divided into three major domains: bacteria, archaea, and eukarya. To construct bacterial or phylogeny, bacterial phylogeny, gene sequencing is a process. It was used. Gene sequencing is used to determine the sequence of genes in any fragment of DNA or RNA. So this process is used to reconstruct the bacterial phylogeny. Bacteria diverge first from archaea and eukaryotic lineage, means they evolved first from the ancestral organism, then archaea and eukaryotic organisms. And the most recent common ancestor of bacteria and archaea, it was believed that probably a hyperthermophile. Hyperthermophile means organism that lived in very high temperature or in hot water springs, and that lived about 2.5 billion to 3.2 billion years ago. This is an ancestral position or phylogenetic position of prokaryotes. You can see that this is a common ancestor from which first of all bacteria were evolved, or they diverge, they get separate from the other organisms. Then the organisms which were left, they evolved into archaea separately and then into eukarya or eukaryotic organisms.
At the broadest level, all life on Earth can be assigned into one of three major groups called domains: domain bacteria, domain archaea, and domain eukarya. While the diversity of life within each of these domains is vast, organisms in the domain share certain characteristics, such as similar cell structures and related sequences of ribosomal RNA. Two of the three domains, domain Bacteria and domain Archaea, contain prokaryotic organisms. These are single-celled organisms that do not have a membrane-bound nucleus. Archaea live in aquatic environments that lack oxygen or are too salty, too hot, or too acidic for most organisms. The ability of some archaea to live in environmental conditions similar to the early Earth gives an indication of the ancient heritage of the domain. As scientists look for life on other planets and moons, they are often looking for evidence of Archaean-like organisms. Bacteria are found almost everywhere, in the water, soil, and atmosphere, as well as on our skin and in our mouths and large intestines. Although some bacteria cause diseases, others can be very beneficial. Many of our drugs are manufactured using bacteria, and bacteria play an important role in the processing of many of our foods. In our bodies, both our digestive and immune systems rely on the action of beneficial bacteria. While archaea and bacteria domains are both prokaryotic, studies of their ribosomal RNA indicate they are not closely related to one another. In addition, their cell walls are considerably different. The bacterial cell wall contains peptidoglycan as well as protein, polysaccharides, and lipids. The archaeal cell wall lacks peptidoglycan, but does contain proteins, polysaccharides, and lipids. The final domain, domain Eukarya, includes the eukaryotes, or organisms that have a membrane-bound nucleus. In addition, most eukaryotes have internal organelles, such as mitochondria and chloroplasts, and, in general, eukaryotic cells are much bigger than the prokaryotes. Domain Eukarya is subdivided into four kingdoms: the protists, plants, fungi, and animals.
In all shapes and sizes, from complex multicellular organisms, such as green plants and animals, to tiny single-celled species, including bacteria, amoeba, and some algae. Scientists believe all life on Earth is descended from a single common ancestor. To help us categorize the species that came afterwards, we group related species. During early evolutionary history, life split into three major domains: bacteria, archaea, and eukaryotes. Prior to this split, early life was constantly evolving. Eventually, the first domain, bacteria, became well-defined about 3.5 billion years ago. Bacteria are usually microscopic, single-celled organisms that are marked by tremendous metabolic innovations. Some bacteria can live in the deep oceans where they feed on sulfur. Bacteria can be both helpful and harmful to other species such as humans. For instance, bacteria in our stomach help us digest food, but other bacteria, such as some strains of Salmonella, can actually cause food poisoning. About 1 billion years after life evolved, domain Archaea formed. Like bacteria, archaea are also single-celled organisms. Archaea weren't discovered or recognized as a separate domain until the 1970s. Some archaea are able to survive in extremely hostile environments, including the hot spring geysers at Yellowstone and the Great Salt Lake. Other archaea can be found in the soil, ponds, and oceans of the world. There are no archaea that hurt humans, as far as we know. Fast forward another 1 billion years or so, and a landmark event occurred. A bacteria seems to have merged with an archaea, and the two work together, eventually becoming a completely interdependent organism. This represents the origin of the eukaryotes. These early eukaryotes gained the ability to protect their DNA with a nucleus, and evolved a cytoskeleton that contributes to the tremendous diversity in shapes in this domain. Eukaryotes include fungi, plants, and animals. Some eukaryotes stole the ability to photosynthesize or get energy from sunlight from bacteria and evolved into a variety of algae that would later go on to become plants. Some early eukaryotes formed colonies and eventually evolved into multicellular organisms such as the plants and animals of today. That's right, you are a eukaryote. It's important to note that while eukaryotes are the only domain to have achieved complex multicellular life forms, that doesn't mean eukaryotes are more evolved than bacteria and archaea. Bacteria and archaea have had billions of years to adapt and survive as single-celled organisms, meaning many of their internal processes are just as advanced, but in different ways.
Now the archaea. Previously they were called archaeobacteria. Archaea means ancient, and archaea is from a Greek word, archeon, means ancient. And in Latin it is also used for prokaryotic cells. Archaean bacteria both are prokaryotic, but still they are different from each other. Their cell wall is different. Cell membrane, its composition is slightly different. DNA sequence is different. And archaea, they live in harsh environment where most of other organisms cannot live. And most of the archaea are methanogenic, means they produce methane gas. And they are also different from bacteria on the basis of their mode of nutrition. And they are also different from bacteria on the basis of ribosomal RNA gene sequence. Now, what are the features of archaea? First of all, as I told you, archaea are different from bacteria on the basis of cell wall composition. Remember, the bacteria, they have cell wall made up of a molecule called peptidoglycan or murine. In case of archaea, they does not contain peptidoglycan, but it is made up of, in most of the cases, polysaccharides, and in some others, or in some archaea, it is made up of protein. As you can see in this figure, plasma membrane, and outside the plasma membrane, this is a cell wall made up of proteins. And there are some archaea which are without any cell wall. Now the cell membrane, composition of cell membrane. Archaea, they contain lipids just like phospholipids of plasma membrane of eukaryotes and prokaryotes or bacterial cell, but their lipid or phospholipid is slightly different, and this unusual lipid allow the archaea to survive at high temperature. And lipids of archaea, they contain glycerol linked to branched hydrocarbons or branched chains of hydrocarbon. Remember, in plasma membrane, phospholipid or lipid bilayer is made up of phospholipid. A phospholipid is a molecule which is made up of glycerol, and fatty acid chains are attached to glycerol. This fatty acid is made up of hydrocarbons, means made up of carbon and hydrogen, generally called hydrocarbons. In case of eukarya and bacteria, the lipids or phospholipids, the fatty acid or hydrocarbons are not branched, but in case of archaea, that fatty acid chains are branched. And this is a figure that shows the difference between the phospholipid of archaea and the phospholipid of bacteria and eukaryotes. As you can see that this is a glycerol to which these two fatty acid chains or hydrocarbons are attached, and these are representing the branches. In this case of archaea, the phospholipid or lipid is branched, or its hydrocarbon is branched, but in case of eukarya and bacteria, this fatty acid chain or hydrocarbons are unbranched. Another difference is that in case of eukaryotes and bacteria, glycerol, it make ester linkage with the fatty acids or hydrocarbons or hydrocarbon tails, but in case of archaea, glycerol have ether linkage with the fatty acids or hydrocarbon. You see this in purple color. This is ether linkage in archaea, and this is ester linkage in case of eukarya and bacteria. And from this structure, I think it is very much clear what is ester linkage and what is ether linkage. Another property of Archaea is methanogenesis, production of methane. The ability to form methane is actually is a type of metabolism which is performed by only some of the Archaea. Methane gas is used in many different purposes in industries and even in the household environment. So methane is produced by Archaea as a result of their metabolic activity. And most of the Archaea, they live in anaerobic environment. Now, what is the mode of nutrition? Most of the Archaea are autotroph, and they use molecular hydrogen and reduce elemental sulfur, carbon dioxide, and water to synthesize their food. So they are chemoautotroph. But there are no photosynthetic Archaea or no photosynthetic Archaeobacteria. Now, the difference or ribosomal RNA sequence in case of Archaea. Remember, the basic difference between the bacteria and Archaea is the difference in the genes that code ribosomal RNA. And it is discovered that the nucleotide number 910 out of 1500 nucleotides present in the gene of ribosomal RNA, at nucleotide number 910, one of the ribosomal RNA is, or the gene or nucleotide is different. And it is different in a following way. You can see that in eubacteria or in bacteria, the sequence of nucleotides of the genes that code ribosomal RNA is AAA, CU, C, AAA. But in Archaea, it is AAA, CUU, AAA. Instead of C, it is U, or C is replaced by U, so that makes a difference. Or on the basis of this, bacteria is different from Archaea.
Have you ever imagined things with a movie trailer voice? You know, that dramatic voice that commands you to listen and be intrigued that shows up on some movie trailers? We think it could actually work great for biology. An example? In a world with no oxygen, one little yeast cell will do fermentation. Okay, tell me that does not make you want to learn at least a little about fermentation. If so, we have a video about that, and the yeast, which is a fungus, which can do it. But you know what would be the best topic for an exciting biology movie trailer? Archaea. Because they are probably some of the most exciting unicellular organisms out there, and many of them can live in extreme environments too. By extreme, I mean think harsh, but then more harsh. Areas with extreme temperatures or high salt levels. Though I should point out, Archaea are not the only extremophiles. Bacteria can be extremophiles too. It's interesting because when I first started teaching, it was common to call Archaea Archaebacteria. But that is an outdated term because Archaea aren't bacteria. Now, they do have some things in common with bacteria. They're both unicellular, they're both prokaryotes, so they have no nucleus, and they also don't have membrane-bound organelles. They both have genetic material and cytoplasm and ribosomes. Both bacteria and Archaea can be classified as autotrophs or heterotrophs, and they both do binary fission to reproduce. Still, bacteria and Archaea are two completely different domains in the three-domain system. That's a pretty big separation. What makes them different? In the three-domain system, you have the domain bacteria, the domain Archaea, and then the domain eukarya, which has all the eukaryotes: plants, animals, fungi, protists. So let's look at the difference between bacteria and Archaea. Membrane structure is very different. Remember that all cells have a membrane. It's really important for homeostasis. If you take a look at our further reading suggestions, you can explore how the chemistry of the cell membrane of the domain Archaea is different. One example is that archaea membranes typically contain ether linkages instead of ester linkages. You would find ester linkages in the membranes of bacteria and eukarya. Some archaea have a lipid monolayer instead of a lipid bilayer. Turns out that a monolayer, as well as some other structural differences of archaea membranes, can be useful for surviving in those extreme environments. You will also find a unique cell wall in archaea. Recall that you will find cell walls in bacteria, and you'll also find them in archaea too. Archaea cell walls can be very diverse, but they don't have this large polymer called peptidoglycan. You will find peptidoglycan in the cell walls of both gram-positive and gram-negative bacteria. While archaea can also technically be gram-positive or gram-negative, most archaea staining gram-negative, archaea don't have the peptidoglycan. Archaea can have pseudopeptidoglycan though, which is a little confusing, but it has a different chemistry to it. Like all organisms, archaea have genetic material. Like bacteria, archaea DNA is generally arranged in a circle, although while bacteria tend to have one origin of replication, archaea tend to have several. When studying the genetics of archaea, it's interesting that now scientists find more similarities between the genetics of eukarya and archaea than they do archaea and bacteria. So overall, these were just some comparisons of archaea with bacteria. Keep in mind that most archaea can't be cultured in the lab. That's because there is a lot unknown about what kind of media they need to grow on, or nutrients or environmental controls in order to grow them in the lab. Although that's a common challenge for quite a few types of bacteria too. Before we go, I want to mention my very favorite.
Now the difference between bacteria and archaea. As we already have discussed, the major differences in the ribosomal RNA sequence that is unique in bacteria. In case of archaea, although it is quite similar to bacteria, but some of the ribosomal RNA sequence also match to eukaryotic ribosomal RNA sequence. Number 2 is RNA polymerase. It is an enzyme which is used by the cell for the synthesis of messenger RNA. This RNA polymerase is small and very simple in case of bacterial cell, but it is complex in archaea just like the eukaryotic cell. Introns, which are the non-coding parts of genes present in the DNA, they are absent in bacteria but are present in some genes of the archaea, and they are also present in the eukaryotic cell. Antibiotic sensitivity, that is their sensitivity to streptomycin, chloramphenicol. It is inhibited by bacteria, but archaea are not inhibited by these different antibiotics. Peptidoglycan in cell wall, it is present in bacteria, but it is absent in archaea, as we have seen that in case of archaea, the cell wall is made up of polysaccharides or proteins, and the lipids in the cell membrane, in case of bacteria, the fatty acid chains or hydrocarbon chains are unbranched, but in case of archaea, the fatty acids or hydrocarbon chains are branched, and their linkage to glycerol is also different. In case of bacteria, glycerol is linked to fatty acids or hydrocarbons by ester linkage, but in case of archaea, glycerol is linked to hydrocarbons by ether linkage. Now the types of archaea, or how most of the archaea inhabits extreme environment. On the basis of this, they are divided into 3 major types. That is methanogens, halophiles, and thermoacidophiles. Methanogens, as the name shows, are methane producing. Halophiles that live in salty environment. Thermoacidophiles in environment with very high temperature and acidic environment. Now, These are the figures which are showing three different types of archaea. This yellow part is the archaea that are living in thermoacidophiles. Methanogen are mostly present in that environment where there is a sewage, and this is a salt lake that is inhabited by halophiles. Now the first is methanogens, or methane producing organism. Methano means methane, gen means producer. So archaea that produce methane. For example, Methanobacterium formicum, they are found in anaerobic environment, means where oxygen is not present. That is in the marshes and in the intestines of human and other organisms. And they produce methane along with ATP from hydrogen gas and carbon dioxide. And this methane gas is also called biogas and is used as a fuel. Number 2 is halophiles that inhabit salt environment. Halo means salt, phile means lover, the organisms that love to live in high salty environment. So they grow on fish and meat which have been heavily salted to keep most bacteria away, like pickles, jams, etc. But in case of halophiles, they grow in environments where salt is applied to the food items to keep the bacteria away, but archaea, they can grow even in that environment. So they require high salt concentration for their growth. For example, Halobacterium, Halobium. Third is thermoacidophiles. Thermoacidophiles are those that love to live in high temperature and in acidic environment. And so they are present in extremely hot and acidic environments such as hot water springs, geysers, submarine thermal vents, means the fissures or cracks present in the seafloor where the heat or thermal heat is produced, and they are also found around volcanoes. For example, Pyrolobus fumari, that is thermoacidophiles.
Archaea includes three groups of bacteria: halophiles, methanogens, and thermoacidophiles. Halophiles are bacteria that live only in habitats with very high salt concentrations, such as brine. Some halophilic bacteria are able to carry out a type of photosynthesis by using the purple pigment Bacterial hodopsin. Methanogens are probably the most abundant of the archaea. They inhabit sewers and swamps, where they use carbon dioxide and hydrogen to produce methane under anaerobic conditions. Methanogens are also found in the alimentary systems of humans and animals. The last group of the archaea is the thermoacidophiles, which inhabit hot, acidic environments. The adaptations of these bacteria to their habitats probably reflect their adaptations to the conditions once found on Earth.
So that was all about archaea. Now the bacteria. Bacterial ecology and diversity. This is a bacterial cell, and the study of bacteria is called bacteriology. And bacteria are ubiquitous, means they are found everywhere, in the soil, in air, in water, everywhere. So this, and show great diversity. Remember the structure of prokaryotes? They have a chromosome in a region that is called nucleoid, which is not bounded by any nuclear membrane, and cytoplasm is bounded by cell membrane. Cell membrane have invaginations, which are the enfolding of plasma membrane, have enzymes for respiration, for cell division, etc. And cell membrane is bounded by cell wall in many cases. Cell wall is also bounded by capsule or slime, and they move with the help of flagellum. They also have fine hair-like extensions which are called villi and fimbriae. Now the occurrence of bacteria. Bacteria, as I told you, are ubiquitous, are present in many different types of habitats, in the air, water, soil, food, even in the bodies of plants, animals, and humans, or on the bodies of plants, animals, and human. Many species, just like Archaea, they are also extremophiles, means they can live in extreme temperature, pressure, pH, saline environment, and other abiotic or non-living environment. Now the major groups of bacteria. Bacteria have been subdivided into major groups on the basis of their cell wall type. Bacteria are divided into two groups on the basis of difference in their cell wall, gram-positive and gram-negative. On the basis of presence of endospores, endospores are the structures which are produced when the conditions or environmental conditions are not favorable for the growth of bacteria. On the basis of metabolism, growth, nutritional characteristics, and physiological characteristics, etc. Now the cyanobacteria, which is also known as blue-green algae, and it was formerly known as blue-green algae. Cyano is a blue pigment present in them, and they are called bacteria because they have prokaryotic cells. So they are more closely related to prokaryotes as compared to eukaryotic algae. Cyanobacteria are photosynthetic bacteria present in the moist environment. They are free-living. Some of them are free-living, some are epiphyte, and some are symbionts. Epiphytes are those organisms that live on trees or on the organs of different plants, maybe on the leaves, stem, etc. They are not dangerous or harmful to the plants on which they are growing. They get their moisture from the air or from the rain or from the dewdrops. And symbiont, I think you are familiar with the term symbiotic or symbionts, two organisms which are mutually living together and both are getting benefit from each other, that are symbionts. And cyanobacteria, they are more related to gram-negative, or the cell wall of cyanobacteria is like gram-negative type of bacteria. Some of the cyanobacteria live individually as unicellular cells, some live in the form of colonies, and some are in the form of filaments. As you can see in these figures, these are unicellular individual organisms in the form of colony, and most of them are in the form of filaments. And the filament in case of cyanobacteria is called trichome. Trichome is a filament made up of cells, and it is covered by a mucilage or a jelly-like substance. So filament which is covered by mucilage, it is called trichome in cyanobacteria. This is the figure of cyanobacteria. This one is nostoc. You see, this is its filament or trichome, and this is Inhabit. Another cyanobacteria or blue-green algae. This is its filament. And cyanobacteria is different from bacteria or eukarya because they have thylakoid-like membranes which are present in the outer region of cytoplasm just inside the cell membrane. Remember thylakoid? Thylakoid are the structures that make up granum, and thylakoid membrane is that site where chlorophyll molecules are attached and where photosynthesis occur. So thylakoid-like structure is not present in bacteria, but thylakoid membranes are present in cyanobacteria, and they are more advanced, or they have structure that is more closely related to eukaryotes. And they also have chlorophyll A that is present in all the eukaryotic photosynthetic organisms. The chlorophyll of bacteria is different. That's why the bacterial chlorophyll is called bacterial chlorophyll, and the eukaryotic chlorophyll is different from the bacterial chlorophyll. And they are of different types. Chlorophyll A is present in almost all the green photosynthetic plants. So it is also present in cyanobacteria. And along with chlorophyll A, which is a green pigment, they have another pigment which is phycobilin. And that is a type of phycocyanin, or a blue pigment. And these cyanobacteria, they use carbon dioxide as a source of carbon, and hydrogen is produced from water, just like eukaryotic organisms, which is different from bacteria. And their photosynthesis also includes, or photosystems. Photosystem is a feature which is present in eukaryotic organisms, in plants. Photosystem is actually a group of chlorophyll molecules that trap sunlight and then start the process of photosynthesis, and it is also called non-cyclic photophosphorylation. So this is more closely related to eukaryotes as compared to bacteria or archaea. And that is important to the evolution of the Earth's oxidizing environment, because it is believed that cyanobacteria-like organisms, they modify the Earth's environment, and they were the organisms that produced free oxygen and made the environment oxidizing in the Earth. So you can see that in this figure, cyanobacteria, just like eukaryotic photosynthetic organisms, they use water and carbon dioxide to synthesize their food, and as a result, free oxygen is produced. This oxygen build-up, or this increases in the environment and change the anaerobic environment of the Earth into aerobic environment. And this is a cross-section, or a transverse section of a cell of cyanobacteria. You can see in the center, this is DNA, which is just like a prokaryotic bacteria or bacterial cell. It is not bounded by any nuclear membrane. These small dots are the ribosomes, which are also 70S ribosomes like the prokaryotes. But these membranes, which are present just inside the cell membrane, these are thylakoid membranes. These membranes, they have chlorophyll, and chlorophyll is also just like the eukaryotic chlorophyll and a blue pigment. So photosynthesis occurs over here in these structures, or in these thylakoid membranes. Now, what is the importance of cyanobacteria? They are important because these are believed to be the organisms that convert the anaerobic environment of Earth to aerobic environment. And many cyanobacteria also fix or convert atmospheric nitrogen into a usable form, that is nitrites and nitrates, which are then used by plants, and the plants then convert it into their proteins, which is then used by animals. So that is the part of nitrogen cycle, because most of the living organisms, they cannot use atmospheric nitrogen to make up their proteins. So this atmospheric nitrogen must be converted into nitrites and nitrates. and that is done by many cyanobacteria in the soil. Many cyanobacteria also have a cell in the filaments. We just have seen a trichome. So this is non-nucleated cell in a filament or in a trichome, and it is believed that this heterocyst is responsible for the fixation of atmospheric nitrogen. This is another figure or a picture of a trichome or a filament. These green cells are the vegetative cells of the filament, but this is without any nucleus, and it is called heterocyst.
Bacteria, which are often commonly referred to as blue-green algae, are an entire phylum of eubacteria. They perform photosynthesis and, unlike other bacteria which perform photosynthesis, release oxygen gas as a waste. It was actually the cyanobacteria which first developed this type of photosynthesis about 2.5 billion years ago, and it is largely due to their action that appreciable amounts of oxygen were added to the atmosphere in fossil Earth. Not only are they phototrophs, which can use sunlight to power their metabolic reactions, they are photoautotrophs, which can utilize this sun's energy to fix carbon in organic molecules. In other words, convert the carbon from carbon dioxide, which is an inorganic molecule, into the carbon found in sugars and other biomolecules. As such, they are producers and very important for food chains. Many filter-feeding organisms depend in part on cyanobacteria. Many protists undergo phagocytosis of cyanobacteria. In addition to being producers, which bring biological carbon into food chains, many cyanobacteria can also fix nitrogen. They have the enzyme nitrogenase, which can then take atmospheric nitrogen gas and convert it into more usable forms such as ammonia and nitrates. They are unusual bacteria in that they are larger than normal, and their photosynthesis is performed on internal membranes known as thylakoid membranes, as opposed to other bacteria which perform photosynthesis using mechanisms on the cell membrane. They utilize chlorophyll and other pigments, which is different from the bacteria chlorophyll used by other photosynthetic bacteria. In addition to being important in food chains, they can be symbiotic with a number of separate organisms. They can have relationships with fungi and form lichens. They can form endosymbiotic relationships with certain protists and animals, including sponges, corals to a degree, and other animals. And it was the early cyanobacteria which formed the first chloroplasts in an endosymbiotic relationship in ancestral plants. And so green plants which perform photosynthesis do so because of the endosymbiotic descendants of cyanobacteria which form the chloroplasts in their leaves. Cyanobacteria can be found in almost every environment: freshwater environments, marine environments, and terrestrial environments. And while many are unicellular, they can be colonial and occur in sheets and chains. Some of these colonies can have some degree of specialization of cells, forming, in addition to the vegetative cells performing photosynthesis, specialized aconites, which can withstand harsher environmental conditions and can be the form they overwinter in, and then the heterocysts in some species which allow them to fix nitrogen.
That was all about cyanobacteria. Now the bacteria, structure of bacteria. All bacterial cells, they have cell walls, cell membrane, cytoplasm, ribosomes, chromatin bodies or DNA, and some structures like capsule, slime, flagella, pili, fimbriae, and granules. They are present in some bacteria but might not be present in some other bacteria. This is again a figure that shows the structure of a bacterial cell. I think now you are very much familiar with this figure. That is a DNA present in nuclide, and this is extra circular DNA that is called plasmid. These are the 70S ribosomes, smaller than the eukaryotic ribosomes, and these are the granules that are mostly the storage granules. Bacteria have a flagellum, which is a locomotory organ, and cytoplasm is bounded by cell membrane, which is just like in composition, like the eukaryotic cells or eukaryotes, which is bounded by cell wall, but the cell wall, its composition is different from the eukaryotic cell wall. It is made up of peptidoglycan or murin, and many bacteria, not all, but many or some bacteria are also covered by capsule or slime, which is present outside the cell wall. Now the structure and composition of bacterial cell envelope. Envelope means that makes outer covering of the bacterial cell. And electron microscopic studies have shown that cell envelope is the outer wrapping or covering of a bacterial cell, which consists of GLYCOCALYX, cell wall, and cell membrane. Cell membrane is sometimes is also considered as a part of bacterial cytoplasm, but it is considered as a part of cell envelope of bacteria, which consists of GLYCOCALYX. GLYCOCALYX may be a capsule or slime, then cell wall and cell membrane. Remember, the GLYCOCALYX, which is maybe made up capsule or slime. What is the difference between capsule and slime? Capsule is tightly bound to the cell wall and gives smooth appearance to the bacterial cell. But in case of slime, it is loosely attached to the cell wall and it gives rough appearance to the bacterial cell. Now, what is the difference in the composition of capsule and slime? As I just told you, capsule is tightly associated with the cell wall and is a condensed layer. It gives a sticky or gummy nature to the bacterial cell, but in case of slime, it is loosely attached to the bacterial cell and gives slimy or sticky nature to the cell, so cell can attach or stick to the substratum or to any object easily with the help of slime. And GLYCOCALYX is made up of polysaccharides. Sometimes it is also made up of proteins. Now, what are the functions of GLYCOCALYX? GLYCOCALYX, that is capsule or slime, it prevents phagocytosis of bacteria by the macrophages. Macrophages are the cells of the immune system that destroy bacterial cells if they enter in the body by phagocytosis. And capsular slime also increases the diameter or effective diameter of a bacterial cell and also make a covering around the cell, due to which they become invisible to the immune system, and they also are prevented from macrophages to be phagocytosed. Capsule, it promotes the attachment of cell to the living as well as to the non-living surfaces. So it results in the formation of a surface on living as well as on non-living surface, and that is called a biofilm. The bacterial cells, they attach to any surface, they increase their number, and they secrete a covering around it that is made up of glycocalyx. So that makes a colony of bacterial cells, maybe of the same species or maybe of different species. So together they make a colony that is called biofilm, covered by glycocalyx. And biofilm are very hard to kill, and that is due to that covering which is made up of glycocalyx. This is a figure that shows biofilm. This is a bacterial cell. It attached to any surface, so increase in number, or many other cells came and then attach at the same site, and together these all cells, they are covered by glycocalyx, so that makes a biofilm. This is another figure of biofilm. You see the bacterial cells, they get attached to the surface, they lose their flagellum, divide and increase their number, and then that colonies of bacterial cell is covered by a slime or a glycocalyx, so makes it very hard to be destroyed. So that is called biofilm. This is another figure. I think now it is very much clear what is biofilm.
Bacteria are typically thought of as unicellular, but in nature, many, if not most bacteria, form specialized surface-attached communities called biofilms. This micrograph depicts a biofilm of bacteria collected from a rock surface in a Yellowstone hot spring. Even in aquatic environments, bacteria are found mainly associated with surfaces. Plaque is a biofilm that forms on teeth. Biofilms play critical roles in microbial pathogenesis and environmental quality, and cost the nation billions of dollars each year in equipment damage, product contamination, and medical infections. Most biofilms in nature are consortia of several species, but in some situations, single-species biofilms can form. For example, the gram-negative bacterium Pseudomonas aeruginosa can form a single-species biofilm in the lungs of cystic fibrosis patients. Single-species biofilms are also readily formed in the laboratory. The first stage of biofilm formation occurs when a specific environmental signal induces a genetic program in free-living planktonic cells. In response to the signal, the planktonic cells begin to attach to nearby surfaces by means of flagella, pili, lipopolysaccharides, or other cell surface appendages. The attached cells begin to coat that surface with an organic monolayer of polysaccharides or glycoproteins, to which more planktonic cells can attach. As the cells enter into biofilm formation, they no longer maintain their flagella, and instead may move along surfaces using a twitching motility that involves the extension and retraction of a specific type of pilus. Ultimately, they stop moving and firmly attach to the surface. As more and more cells bind to the surface and divide, they form microcolonies and can begin to communicate with each other by sending and receiving chemical signals in a process called quorum sensing. These chemical signal molecules are continually made and secreted by individual cells. Once the population reaches a certain number, analogous to an organizational quorum, the chemical signal reaches a specific concentration that the cells can sense. This concentration triggers genetically regulated changes that cause cells to bind tenaciously to the substrate and to each other. Quorum sensing may influence the amount of extracellular matrix produced by the cells. This thick, slimy material consists of polysaccharide polymers called exopolysaccharides and entrapped organic and inorganic materials. The extracellular matrix protects the cells from damage from environmental assaults. Quorum sensing serves the biofilm in many ways, such as by increasing the resistance of biofilms to antibiotics. Quorum sensing in Pseudomonas aeruginosa plays a role in the maturation of the biofilm into complex three-dimensional forms, such as columns and streamers, forming channels through which nutrients flow. Sessile cells in a biofilm chemically talk to each other in order to build microcolonies and keep water channels open. Biofilms tend to form where nutrients are plentiful and may begin to dissolve when nutrients become limiting. Little is known about how a biofilm dissolves, although the process is thought to be triggered by starvation. Pseudomonas aeruginosa produces an enzyme that can strip away the exopolysaccharides. Cells leaving the biofilm revert to their flagellated planktonic forms and can swim to new locations where nutrients are more plentiful. Pseudomonas aeruginosa can be medically devastating. It forms biofilms in susceptible individuals, such as in the lungs of patients with cystic fibrosis, and on medical implants.
Now the second component of bacterial cell envelope, that is cell wall. It is present outside the cell membrane, and in bacteria it is made up of peptidoglycan, which is a large molecule and is also called murin. In case of gram-negative, the peptidoglycan is covered by another layer which is made up of lipoproteins or lipopolysaccharides, but this is only in the case of gram-negative bacteria. Gram-positive bacteria, they have only a layer that is made up of peptidoglycan. And peptidoglycan, it provides, just like the cell wall in higher plants, a structure which supports the cell and gives a characteristic shape to the cell. This is a figure that shows peptidoglycan. You can see that this is a large molecule that makes the cell wall of both gram-positive and gram-negative, but in case of gram-negative, this peptidoglycan is covered by another layer which is made up of lipopolysaccharides or lipoproteins. This is another figure that shows peptidoglycan. And this figure shows the difference between gram-positive and gram-negative. Actually, they are called gram-positive and gram-negative on the basis of a staining that was for the first time proposed by a scientist, Gram. Gram-positive are those which stain with violet color, and gram-negative, they get pink stain. Actually, that is difference. That is on the basis of difference in their cell wall, they get two different types of stain. As you can see in this figure, during the staining process, first of all, the crystal violet color is, or stain is applied to the cells which are placed on this slide, and then iodine is used to stain the bacterial cells, and then the stains is washed with alcohol. And actually the purpose of washing is to get rid of extra stain. Gram-positive, as they are made up of peptidoglycan and they have no outer covering, so they get that violet stain and they appear violet in color under the microscope. But in case of gram-negative, as they have outer covering which is made up of lipopolysaccharides and lipoprotein, when after iodine staining, the slide is washed with alcohol. The alcohol wash away the lipoprotein and lipopolysaccharide layer, so that violet color is retained up to that lipoprotein and polysaccharide layer, and it do not penetrate up to peptidoglycan, which is present inside that outer layer. So when the cells are treated with the second stain, that is pink in color, saffron, so that is gram-negative, which are at the stage without any color or without any stain, so they get this pink color, so they appear pink in color under the microscope. And these gram-positive, as they already had violet stain, so they do not get second stain that is pink in color. So on the basis of difference in the cell wall of gram-positive and gram-negative, we classify bacteria into two major groups, gram-positive and gram-negative. In case of gram-negative, as you can see that this is cell membrane. Outside the cell membrane, there is a thin layer of peptidoglycan, and peptidoglycan is covered by another layer which is made up of lipopolysaccharides or maybe lipoproteins. And this outer membrane also have porin. Remember porin? It is a protein which is used for the transport of certain molecules. And another difference in the gram-positive and gram-negative is that there is a space between the peptidoglycan layer and cell membrane. This space is called periplasmic space. This space is absent in gram-positive bacteria. So, in case of gram-negative, there is an outer membrane outside the peptidoglycan which is not present in gram-positive. This is a gram-positive bacterial cell wall. You see, this is a cell membrane, and cell membrane is bounded by a thick layer of peptidoglycan. And another difference is that gram-positive bacteria also have the fibers of teichoic acid. As you can see over here, these fiber-like structures, these are the teichoic acid which are protruding from the surface of peptidoglycan of gram-positive bacteria. This is a figure that shows the difference between the gram-positive and the gram-negative bacteria. As you can see that this yellow color layer is representing the cell membrane, and this brown color is representing peptidoglycan, and these fibers or these structures are the teichoic acid which are protruding from the surface of peptidoglycan of gram-positive. You can see that in case of gram-positive, the peptidoglycan layer is very thick, and there is no space between the peptidoglycan and cell membrane, means there is no periplasmic space. But in case of gram-negative, you see that this yellow part is a cell membrane. Then outside the cell membrane, there is a thin layer of peptidoglycan, which is very thin as compared to gram-positive. And outside this thin layer of peptidoglycan, there is another layer which is made up of lipopolysaccharides or lipoproteins, and they also have porin, which is a protein that is used for the transport of certain molecules. Another difference is that there is a periplasmic space present in gram-negative bacteria between the cell membrane and peptidoglycan. Periplasmic space consists of certain enzymes like beta-lactamases, and these are used to destroy certain drugs or certain antibiotics. This table is again showing a difference between gram-positive and gram-negative cell wall. As you can see that in case of gram-positive, the number of layers outside the cell membrane is only one, that is made up of peptidoglycan. But in case of gram-negative, it consists of two layers. And in case of gram-negative, the peptidoglycan is very thick, about 20 to 80 nanometer. But in case of gram-negative, it is thin, about 8 to 10 nanometer. And in case of gram-positive, there is no outer membrane or no membrane outside the peptidoglycan. But in case of gram-negative, the outer layer, which is made up of proteins or polysaccharides, is present outside the peptidoglycan. And periplasmic space in most of the gram-positive bacteria is absent, present only in some gram-positive. But in case of gram-negative, it is present in all gram-negative bacteria. And the composition of outer layer of gram-positive is peptidoglycan. It also consists of teichoic acid and lipoteichoic acid. But in case of gram-negative, the outer membrane is made up of lipopolysaccharides and lipoproteins, And inside that outer layer there is a peptidoglycan. And porine proteins, they are absent in case of gram-positive, but they are present in case of gram-negative. And lipid content in case of gram-positive is very less, but it is more, or it is higher in case of gram-negative, as lipids are also the part of outer layer which is present outside the peptidoglycan layer. So the peptidoglycan is more in case of gram-positive and less in case of gram-negative. And the permeability of molecules in case of gram-positive is molecules can easily and they penetrate more as compared to gram-negative, where the membrane is not that much permeable. And in case of gram-negative, it is resistant to more molecules as compared to gram-positive. Gram-positive is less resistant, but gram-negative is more resistant. As I just told you, that periplasmic space which is present in most of the gram-negative bacteria, it have enzymes which destroy antibiotics or drugs. Now the diversity in size of bacteria. Bacteria are of many different shapes and sizes. The smallest bacteria, it belongs to the group called Phytoplasma, and these are the bacteria that infect plants. They are spherical, as you can see in this figure, and their approximate diameter is 100 to 200 nanometer. That is 0.1 to 0.2 micrometer in diameter. And this figure, as you can see, that these are spherical bacteria, belong to a group Phytoplasma, and most of them infect plants. Then Escherichia coli is a bacteria. Generally it is called E. coli. They are rod-shaped, of about 7 micrometer long and 1.8 micrometer in diameter. And they are also used to test water for fecal contamination, because they are present in the feces of some organisms which are infected by E. coli. They are long as compared to narrow. And another type is Spirochaeta plicatellis, as you can see in this figure. These are spiral-shaped bacteria, which is about 250 micrometer long and 0.75 micrometer thin. And for comparison, human red blood cell is 8 micrometer in diameter, so it is longer than the red blood cell. Another one is a giant bacteria that is called giant bacteria because it can be seen with the naked eye, and its type that is known as Epulopiscine fischelsoni. It lives in the gut of brown surgeonfish that is found in the Red Sea, and its diameter is about 80 micrometer thick, and it is more than 600 micrometer long. And due to this size, it is visible even to the naked eye. It can be seen with the naked eye without any microscope. That's why it is called giant bacteria. That's all about today's lecture. Thank you very much.